Hi, everyone. I'm Sophia. I work under Dr. Pete Buston, where we are a marine evolutionary ecology lab that focuses on the mutualistic benefit of anemones and anemone fish. But my specific project is finding home for Nemo. Can clownfish protect their anemones from rising heat? So first, I want to go into a little bit of background before I go into the bulk of my experiment. And that first starts with climate change. So as we know, climate change is significantly impacting our ecosystems. And with that, we have rising temperatures within our oceans. This leads to things like habitat bleaching. I'm sure we all know about our coral reefs, but they're experiencing a significant impact of coral bleaching, which is causing death for a lot of species. This rising temperature impacts species interactions, as well as a change behavior for these marine species. Now, these three driving examples are leading to the idea of mutualism. So this is just a symbiosis that's beneficial to both organisms involved. And one of the most common and well-studied mutualistic relationships in marine ecosystems is that between coral and zooxanthellae. Zooxanthellae is just it's a scientific way of saying algal symbionts. So those are what give coral that really beautiful color. But what's really understudied is the mutualistic benefits and relationship between anemones and anemone fish. Anemones and anemone fish are known to give each other benefits like nutrient exchange, protection from predators, as well as increased water flow. But what I really want to look at is if these types of benefits break down during heat stress events, or if they can help provide some sort of buffer to the anemones. So one of my main questions is if mutualism can help buffer the bleaching effects of anemones, as well as what role do the anemone fish play in mitigating this response. So I wanted to show this really cute video of the clownfish and the anemones that I work with. And not only are they super cute, but also you can visually see the type of mutualistic relationship that these guys have. Unknown fact, clownfish are terrible swimmers, and they'll often spend most of their life within their anemones. But this interaction is super beneficial for the anemones involved because of that physical touch. So the two species that you saw there, the first one is E. quadricolor. This is known as a bubble tip anemone. And then the next one is A. percula. So this is known as the true clownfish. So yeah, I get to work with Nemo every day. And next, going into our setup. So again, these are our two species, but instead of working with adult clownfish, which can live up to like 35 years, I got to work with juveniles, which were around 1 month old. So our setup include either an anemone or an anemone with anemone fish. And then it included these 96 microcosms. So these are these small ecosystems. This is a picture of what one of our basins look like. So I had a total of 4 basins, 2 being controlled temperature and 2 being heated temperature. But these basins can include things like an anemone, and an anemone fish, or maybe just 2 anemone fish. So I know I just talked about 2 basins being control and 2 basins being heat, but what does this actually look like? So on our x-axis, we have our weeks. So this was an 8-week experimental period. And on our y-axis, we have our mean temperature. So during this 8-week period, out of 4 weeks, they were ramped up to 33 degrees Celsius, which is what they're experiencing in their natural environment, and then ramped back down because we know that heat stress events don't last for an entire month to 2 months, but usually there are fluctuations of temperature. So this was both for just the anemones and the anemone fish and the anemones, and then they had their counterparts of controlled, which were maintained at a constant 27 degrees Celsius. So the 3 variables that I measured were growth, distance, and color. So with growth, we know that bleached anemones or stressed anemones tend to close more, which is pictured at the top. This is a picture of one of our bleached anemones, and you can tell compared to the bottom, its tentacles just aren't as expanded as they should be. So we wanted to see what the expansion rate was, so whether they expanded or contracted, and whether this was some sort of stress response. So I used ImageJ, which is just a coding software, where I used an ellipse tool, which is pictured at the bottom right, to measure the area or the change in area over our 8-week time period, and to see if these anemone fish actually influenced their contraction and expansion rate. The next thing I looked at was distance. So we know that bleached anemones or stressed anemones tend to move more as an escape response. So what I did is I found the pinpoint of their oral cavity or their mouth, and I measured the distance every day. So this is just an example. So I would measure, like, if this anemone in the microcosm moved here to here, I would measure that distance. And the last thing I measured was color. So we obviously know, just like corals, bleached anemones tend to have that really bright white color. But in order to actually analyze this, we pulled the red, green, and blue values, which are just like how red, green, and blue something is. And then we used equations to find the luminosity, saturation, and hue. So that's just how bright or how orange something is for our case. And this was used for Photoshop, so we made sure everything was white balanced, so it was exactly the same. And then it was run through MATLAB to pull out those red, green, and blue values. And then all of this was statistically analyzed using R, which was really hard, but I did it. And before I go into my results, I want to talk about 3 periods of time that we use to calculate everything. So it's one thing to just look over the change in the 8-week time period, but we wanted to see if there was any form of resistance from the heat stress, recovery after the heat stress, and resilience over that total time period. Okay, yeah, so resistance, anyway, sorry about that. Resistance is known as our middle minus our initial, or our week 4 minus our week 1. And then our recovery is gonna be our final minus our middle, so our week 8 minus our week 4. And that resilience, like I was saying before, is that total time period. So to visually look at it, if you guys remember the heat ramp that I showed you guys before, our resilience is our total time, resistance is that resistance to that heat response, and recovery is after that stressor event. So really complicated graph, but I really want you guys to look at is the resilience time period. So that's your far right. But what we have on our x-axis is our organismal treatment. So that's anemone versus fish and anemone. And on our y-axis, we have our area or our change in area. So here what you can see is we have a significant result between our control versus our heat, so our blue boxes versus our red boxes. And this is during our resistance time period, so our week 4 minus our week 1, and our resilience time period, so for our total week. And this is basically saying that there seems to be a significant change in area for our heat versus our control, and this is really driven during our resistance phase. It is also important to note that there's no significance between organismal treatment or an interaction between the heat and controlled and the organismal treatment. So basically meaning that the presence of an anemone fish didn't have any impact on the stress response of an anemone during a heat stress event. So again, this is like pretty similar to the graph I just showed before, but our change is gonna be in our y-axis. So this is measuring our distance. And unlike the graph that I previously showed, there's a significance with our recovery and resilience instead of our resistance and recovery. But this also significance is only between our heat and our controlled environments, and there's no significant effect of the anemone fish on an anemone. And again, the resilience is really driven by this recovery period, so after our heat stressor event. So although it's disappointing to know that based on our results that the presence of an anemone fish doesn't seem to have an impact on the mutualistic relationship between the two during stressor events, it's also important to note that we were working with juvenile fish and really small anemones. In the wild, this is not the case. Anemone fish can live up to 35 years old, so they're obviously a lot bigger. They get up to 18 centimeters. The ones I was working with were about 3 centimeters on average. And then the anemones can also get up to 30 centimeters, but the ones I was working with were about like 5 centimeters in diameter, so obviously a change. So the lab conditions were just very different. Also, this mutualistic relationship did not evolve in a time where these heat stressor events were normal. What we're seeing is anthropogenic change to our oceans that's significantly impacting our ecosystems, and this is not something that our organisms were ready for. But in the future, I am going to the field this summer to continue my research in the field. I get to go to Papua New Guinea, which is really fun, but I get to see these organisms in action, not only in a lab setting, but in the environment. Thank you all so much, and I'll take any questions.
Hi, Sophie. Thank you for that presentation. My question was about the comparison between the lab conditions and the field conditions and what your research this summer will include.
Yeah, so I'm working with all the PhD students in our lab, and I'm on 2 projects. One is larval dispersal, which really has nothing to do with what I'm doing now. And the next one is bleaching effects. So in Papua New Guinea, where these fish and anemones are found, we're having increased bleaching. It's around, I think it's like 80% of what we're seeing totally bleached. So the PhD student that I was working with previously focused on before they were bleached, so we're seeing behavioral effects, so videotaping how much time they spend in the anemone versus out of the anemone, and if there's any sort of nutrient balance as well. And then within the lab, it's just very controlled. We try to replicate as much as we could that's happening in the wild, but things like the anemones and the anemone fish were fed every single day, so there wasn't that normal nutrient balance that we're normally seeing in the wild, and the fact that we're just working with younger anemones, younger anemone fish, that's just not replicated or what we're seeing in the oceans.
Hi, phenomenal presentation, Soph. Quick question, you mentioned 80% of what you guys are seeing out there is bleached. Is there any studies going on the 20% that aren't? Any characteristic or genetic makeup that's possibly making them more resistant?
Yeah, so that's what one of the PhD students are doing. They're seeing if there's a difference between those anemones that have bleached and those who haven't bleached. And this is both from a mutualistic relationship, so the anemones versus the anemone fish, but also if this is impacting any larval dispersal. So those anemone fish that are born, they go into the pelagic sea for a long time, but then they return to the same anemones or around the same reefs that they were born at. So there's also studies being done if this is impacted at all, and if it also impacts their social interactions, because clownfish have a really interesting social hierarchy. So if the bleached anemones also impact that, because you can see, like, even from this picture, it's really obvious where the anemone fish is for the bleached one, but not as obvious for the other. So if this also impacts predation.
Time for one more question. I saw Caroline.
Great presentation. My question was basically your research this summer. Obviously, we talked about one anemone. I'm just assuming that there's many other anemones. Will you be focusing your research on just this one, or are you going to be comparing all of them and just pulling research from that?
Yeah, so I'll mainly work with E. quadricolor, which again is the bubble tip anemone, but people in our lab also work with the giant sea anemone, which is mainly found in the Red Sea. But I'm specifically just focusing on what's going on in the Pacific and what's going on between the interaction of the true clownfish and the anemone.